This is a production of Cornell University.
So I'm going to be talking about developing a little bit about developing multi-use naked barley for New York. First, going to focus on what naked barley is, what are its potential uses in a future market, some of the challenges of naked organic barley, and I'm going to do a little bit of focus on some results so far in terms of wheat competitivity, and if there's time, I will look at show you some of the protein and yield data from the first year. So what is naked barley? It's simply barley without the hull. And so what that means is when barley is threshed, when harvested, the hull falls off, similar fashion to wheat. Compare that to malting barley where the hull remains on the grain. Some examples right here are Copeland and Full Pint, which are hulled barley, and then Buck, which is hullless or naked, in which the hull falls off. This gene for hullless barley is characterized by the NUD gene on chromosome 7HL. It's recessive in its loss of function single point mutation. And so in theory, it's quite easy to breed out of after a couple selfing generations. However, there are some modifiers which make it a little bit more difficult to make truly naked sometimes. The big advantage of hullless barley or naked barley compared to hulled barley is that without the hull, you don't have to pearl it, and it can be useful for food purposes. In order to determine the potential for naked multi-use organic barley, we're kind of basing our potential success off these two movements or sources of increased markets. The first being the craft brewing movement. As Dan mentioned in seminar last Tuesday, there's been increased movement for using naked malted barley, particularly in New York. The New York Farm Brewery Bill has incentivized local brewers and maltsters to source their ingredients from New York. That, and combined with the organic grain market, which there's been an increase in demand for novelty food grains, and with previous work of value-added grains with Mark, Lisa Kissing-Kusek, and June Russell, have been really instrumental in developing a market for organic grains. So hopefully with the combination of the malting and brewing movement and the organic grains, we can replicate the success with multi-use organic barley. So what are the potential benefits specifically? As I previously mentioned, the characteristic of no hull is its main selling point. Health benefits such as beta-glucan and protein levels compared to wheat. But mainly, we're really focused as an alternative crop for organic farmers. And so some of these characteristics that we hope that organic farmers can sell to are malting. And if malting doesn't work, we can do food. And if food doesn't work, we can go for the feed market. However, the feed market can be a bit dismal, but perhaps with organics, there'll be a better market compared to the conventional counterpart. So unfortunately, with naked barley, there's a lot of challenges, and I'll start with the agronomic ones. Of course, we have to worry about yield with barley, winter kill for the winters, and lodging. And then we have to worry about environmental factors that barley is highly sensitive to, disease resistance, scald and powdery mildew for the winters, leaf rust, and spot blotch for the springs, and of course, Fusarium head blight. For malting and food, we have to worry about aesthetic traits. Is the barley going to be marketable? For example, if you have a purple barley, that's going to be definitely more marketable than, say, a conventional white barley. Protein and beta-glucan. And more specifically within the protein beta-glucan, your food barleys will probably, where you might want your food barleys to have a higher level and malting to have a bit lower level. And then, of course, in New York, we have the pre-harvest sprouting issue, which was really a problem this year, where the grain germinates in the field. And so we want to avoid that. And in addition, with disease resistance, we're concerned about the Don being part of the grain. And then there's the organic specific challenges. With yield, you have different management concerning the grain, fertilizer applications and whatnot, certified organic land, what nutrients are available for that, disease resistance, you have limited amount of fungicides that you can use. And for some seed-borne diseases, there is currently no solution. And of course, weed competitivity. And so put all these traits together, you have quite a bit to consider. And so while we hope to make gains in all of these, making idealized gains in all of these won't necessarily be possible. So for what I'm going to be talking about here, I'm going to be focusing on these three traits: protein, weed compatibility, and yield. So for the project, these are the three main approaches. We have the marketing, to market the grain as something that people should buy, which is kind of putting the cart in front of the horse a little bit with this breeding project. Education, we have a barley composite sent to a local middle school, so they get to learn about aspects of barley or any genetic concepts the teacher might want to talk to them about, and then the research component, which is what I'm here for. So we have both a diversity panel and a regional panel, regional trials. I wanted to talk about the diversity panel. However, we did get some good data on height, head of date disease, but due to the inclement weather, short amount of time, and it being a diversity panel, the seed phenotypes were not that great. So I'll avoid talk about that for now. And then, of course, regional trials. So for the regional field design and management, our entries included some release varieties, including bucket streaker, that we've grown out for Grow New York City already. Some hauled checks, selections from Oregon State based on previous diversity panels they've conducted and breeding efforts, and some naked lines that have been grown here for the past 2 years. For the field design for these trials, we had 25 entries for the winter, 20 for the spring. Each one is 3 replicates, randomized complete block design. For the locations, for the winters, we had only one location at Freeville, and for the springs, we had two locations for the springs, one also at Freeville and one at Musgrave. And in regards to management practices relevant for these phenotypes, we did do tine weeding. As much as I would like, as much as we might be able to get a better weed percent management without tine weeding, breeding for weed compatibility is supposed to complement organic practices, not replace them. And of course, organic fertilizer rates are going to affect yield and protein somewhat. So for weed compatibility, attempting to find what characteristics are related to yield are quite difficult. They're easily confounded with environment. Things such as previous year rotation of what crop was there before it, just random factors that result in weed seed dispersal make this a very hard phenotype to measure. So pretty much what I've done is trying to do like a shotgun approach and identify certain barley traits and weed measurements to see how well, if any of them correlate to yield, and we can use them as evidence of an entry having a higher weed compatibility compared to others. So some of these traits that I phenotyped included stand counts in October and November 2017, early vigor, where 1 is like the plant's not even there, and 9, the plant completely fills out the plot. In May and June, I did weed barley percentages, and I measured that by using this PVC 0.5 meter squared plot by throwing in a random section, and then seeing what percent within that plot is either barley, weeds, or neither, so it would be ground. And then did that twice for each plot, and then did it for all of them. And then recorded plant form, where 1 is prostrate, so the barley spreads out, and 5 is erect. And recorded these values for weed and barley plant form 3 times every 2 weeks. So these were the results of the weeds for the winter regional. This bar graph showcases the varieties that had the greatest differentiation for weeds. Unfortunately, a lot of the unfortunately, there wasn't a lot of weeds in winter, or at least they weren't very correlated with yield, most likely due to the dry weather conditions. And or it could be that all these barley varieties may have some effect in weed suppression, but between barleys and other plants, we haven't grown those yet, so it would be hard to say. So these were some of the correlations for these traits that I measured. Overall, plant form and percent weeds wasn't very highly correlated. Barley, not surprisingly, if there's more barley given in the plot, it's going to have a higher correlation. Stand counts and vigor had some correlation, but nothing too promising. But it's important to keep in mind this is only the first year. And so we're going to need to do this over multiple years to get multiple weather conditions that will be more desirable for weeds to grow, and maybe not so much for barley, to see whether these barley will truly outcompete the weeds as in a trait. So we did the same thing for the springs, locations at Freeville and Musgrave. Did weed same traits. The only thing that I did add is that I did weed counts when I did the weed barley percentages as a proxy to see, well, maybe if we account for the type of weeds or the size of the weeds, maybe that will have a greater correlation. And recorded those 3 times every 2 weeks as well. Unfortunately, that was even worse due to the drought, due to the severely localized drought in Musgrave, and the fact that these were planted very late on May 25th. There wasn't many weeds in the first place because even weeds don't like dry weather. Lots of ground space as well because the barley's had a hard time growing in this environment. So unfortunately, we weren't able to get much weed measurement data. Now these values of barley, again, are highly correlated. Surprisingly, some of the weed measurements were more highly correlated. However, it's really hard to say whether it was the barley that suppressed them or if it was more rather they filled in the void of the barley just not growing in the first place. Freeville, we had a little better success, little better weed pressure than Musgrave or the winter location, but even then with the drought, weed conditions still, the weather conditions still impeded the growth of weeds. Looking at some of these correlations, again, our 2 highest ones were barley, and for this one, it was a vigor rating. In the future, for this trait, it'll probably be better to measure barley at certain stages of the plant where barley is more highly correlated if we want to determine whether these can grow faster. Either that, or we're going to have to do a better job of accounting for the weeds. And so for that, we would have to do more of an experiment where we would have to seed the ground with a certain population of weeds that are relevant to that stage in development of the barley. So so these are questions I would hope to answer. How much time do I have? 2 minutes, okay. So for overall yield performance of the winners, they did quite well. This is the statistical model I used to fit responses yield. It didn't put any fixed effects in, as any of the fixed effects tried had no significance. The random effect was with entry modeled as such. And for the error variance, you did a row column spatial correction where the diagonals represent the error for each plot, and the off diagonals represent the distance correlated errors between the plots. And overall, these yields did quite well, but it's important to consider that this is only the first year. A lot of these are not specialty selected 2-row malting, so a lot of the energy for the plant can be focused more on the grain yield as opposed to other traits that you would select for malting. So that may be a contribution to why these are a bit higher. And then these are for the springs. The only thing that was different is that I added a fixed effect for location. And as expected, these did not do so well. Springs had a very hard time this year with unfavorable weather conditions. And even between the dismal conditions at Musgrave and Freeville being marginally better, the distribution of these scores are still quite narrow. So the weather certainly played an effect for that. I also did protein, took a 20-gram to phenotype protein, took a 20-gram sample from each plot. Samples are analyzed using a FOSS-NIR system, using least squares regression. And depending if the barley was a hull-less or a hull check, they're calibrated on separate files, because hull calibration files do not help calibrate hull-less ones or naked ones. So for overall protein performance, the same model, use the same model as yield, only with responsive protein instead. Overall, for the winters, they did quite well in terms of our desired range for what it should be for protein. However, this is only 1 year, and we'll still need to evaluate these across better years to get a better picture of how these will respond in terms of protein ranges. And we have not phenotyped for beta-glucan yet, but that should be another factor in determining whether these might have some potential malt quality. For the springs, these also didn't do well in that regard either. Same model as the previous one, except adding the fixed effect of location. Due to the weather, protein values tend to be much higher in barley when there's not enough water or there's a lot of drought. And so hopefully next year we'll have a better accurate depiction of these spring barleys with better weather conditions. So future plans in terms of multi-use organic barley, with the genomic data of the diversity panel, I hope to combine with the phenotypes, have some potential information for some genome-wide association studies, and perhaps examine different methods of measuring the rate of growth of barley. And with that, acknowledgements, I'd like to thank my community members, Mark, Gary, and Sam Alkane, on the entire Soils Lab Group, funding sources, the Annick Lab Group, and Synapsis. Any questions? Yep.
When you were measuring protein, you showed a picture of your seeds germinated. Is that because when you measure protein, it's post-germination, or is that from the field?
I apologize for that. That was just a general naked barley picture. That was not germinated. I apologize for putting that one up there. It was not relevant to the protein. My apologies.
But typically, are you measuring protein from material from the field or post?
From the field. Directly take a plot sample from plots. Yep. After they're harvested and weighed, of course.
For the weed competitiveness, are you concerned with which weeds they are, or in this more locally going forward?
Yeah. So I didn't put that data in here, but I did take note of which ones they were, and to account for that, I did take kind of the size. But yeah, I have been taking notes on what kind of weeds are present. So if I were to do a seeding experiment, those are the weeds that are used. Generally, it's a lot of lambs for the winters. You have to worry about henbit and chickweed, and for the springs, you have to worry about pigweed and lamb's quarters more. So.
Did you mean to use a flat correlation for the errors between plots, or was that supposed to be an AR1?
That was supposed to be an AR1. Yeah. Yep. Okay. Thank you.
Yes. Thank you, Karl.
This has been a production of Cornell University, on the web at cornell.edu.